is Dr. Monica Kinde, and we're very excited to have one of our Joplin faculty here at Research Day today representing the campus. And she'll be talking to us about fluorine NMR quantification of isoflurane binding to the voltage-gated sodium channel NaChBac.
Awesome, thank you. I'd like to extend my thanks to the symposium committee for allowing me to come up here and representing the Joplin campus. I'm going to talk some basic science to you guys, so I hope you're not too bored. This presentation includes some work I did during my postdoctoral experience at the University of Pittsburgh in the departments of anesthesiology and structural biology. I'm going to try to cram as much info as I can in 15 minutes, but if you'd like a little bit more comprehensive view of this story I'm going to tell you, I implore you to go to this citation here because it was published last November in the Proceedings of the National Academy of Science. So some of the techniques and the general topic I'm going to talk about, I'm going to consider as I develop my own research plan down in KCU Joplin. So this work represents a supremely collaborative effort that was funded by an NIH PO1, and a PO1 is a research program project. It's broadly based, multidisciplinary, and often long-term, and it has a very specific major objective. So our team at the University of Pittsburgh were a combination protein and animal group. So a lot of the insights that we made from our protein structural analysis, we were able to, in keeping with the theme of today, directly translate it to a more animal model. So, and then we have our MD team at Temple and Drexel. They did a lot of our in silico work, and then our the main PI of this PO1 was Dr. Roderick Eckenhoff at Pennsylvania University. And so we're all multi-institutions operating under the umbrella of the very generous NIH. So this work attempts to answer or add understanding to one overlying question, and that question may come as a surprise to you. Well, maybe not the clinicians in the audience, but to a bright, kind of young, a bright-eyed young biochemist just starting her postdoc. It was quite a surprise to me that this question even needed to be answered still. And that question is, what is the mechanism of anesthesia? So how does anesthesia work? Anesthesia has been readily used since 1842, so it's generally pretty surprising that this question is still unanswered. However, it's generally accepted that general anesthetics exert their effects by modulating the activity of membrane proteins in the neuronal membrane. So to more closely study the specific interactions between these receptors and anesthetics, we employ a variety of techniques. So we have this central question, what is the mechanism of anesthesia? And we use a variety of biophysical techniques. We employ molecular biology techniques, protein expression, in silico methods, including molecular dynamic simulations, and electrophysiology. So most of the work that I'm going to talk about today uses NMR, protein expression, MD, and 2-electrode voltage clamp electrophysiology. So I assume most everybody in this room at one point in their careers has suffered through organic chemistry, right? But you might be asking, so you should know what NMR is, but as a refresher, NMR is nuclear magnetic resonance. So let me ask, how many of you in the audience have ever had an MRI? Anybody? Congratulations, you have been an NMR sample. So the same way that an MRI works is the way that an NMR works. So it just uses specifically tuned radio frequency to obtain detailed structural pictures. In an MRI, it's a structure of a human body system. In an NMR, it's a structure of a simple organic molecule or a very large complex membrane protein. So NaChBac is a bacterial homologue of the eukaryotic voltage-gated sodium channel, and it shares a lot of structural and pharmacological properties with its eukaryotic counterpart. So this is just an example of a human voltage-gated sodium channel, and you can see, comparing NaChBac structure to the human channel, there's a large degree of structural homology. Voltage-gated sodium channels are transmembrane proteins that form ion channels that are activated by changes in the electrical membrane potential. And changes in the membrane potential alters the conformation of the channel protein, which then regulates the opening and closing of the channel itself. So voltage-gated sodium channels allow rapid and coordinated depolarization in response to triggering voltage changes. And they are found along the axon and at the synapse, and they directionally propagate electrical signals. So NaChBac is a homotetramer, meaning it's composed of 4 subunits that you can see are colored in different colors here. And it is organized around a central ion pore. So if we turn this on its side, and we're looking at it from the cytoplasmic region, you can easily see the central ion pore right there. So each subunit contains 6 membrane-spanning segments designated S1 through S6 that take the form of alpha helices. So the pore domain contains helices S5, S6, and the pore helix, and this is the pore domain. The voltage sensing domain is comprised of S1 through S4 and serves as kind of the voltage sensing region. And the reason that we use NaChBac is it can be readily expressed in bacterial strains like E. coli, and we're allowed to get a large, very pure quantity of NaChBac that allows us to do these structural studies. We can say that, but then at the end of the day, we still have to justify using a prokaryotic system to the NIH, which is why it's nice at the research group at the University of Pittsburgh, we're immediately able to take these same proteins and look at them in an animal model. As I mentioned, it is generally accepted that anesthetics have specific molecular targets, and these targets are of several varieties. We have ligand-gated ion channels, voltage-gated ion channels, and G-coupled protein receptors. Admittedly, a majority of my work at the University of Pittsburgh was on cis-loop receptors. And so cis-loop receptors are found predominantly at the postsynaptic neuron. However, voltage-gated ion channels can be localized at both the pre- and the postsynaptic terminals. And they either augment neurotransmitter release at the presynaptic terminal, or they contribute to postsynaptic neuronal depolarization. So as you can see in this table right here, there's been a large variety of electrophysiology and structural studies of various anesthetics on these ligand-gated ion channels. However, it's kind of glaringly obvious how very little structural studies have been done on voltage-gated channels. So in particular, it is assumed or hypothesized that inhaled anesthetics in particular induce changes in voltage-gated channels that induce the anesthetic response. So despite knowing that isoflurane in particular acts on voltage-gated sodium channels, it's not exactly time or resource or cost effective to go in kind of willy-nilly and start doing all these structural studies without having some sort of guidance to guide us into structural regions of interest. So we looked to our in silico methods to do that, because ultimately in silico is the cheapest method you can get, really. So our groups at Drexel and Temple did what's called molecular dynamic simulations, where they took NaChBac, equilibrated it in a lipid bilayer, and did molecular flooding experiments. So they flooded this NaChBac with thousands of isoflurane molecules and allowed the simulation to equilibrate over a certain length of time. And at the end of the simulation, isoflurane tends to bind at specific regions. And they discovered 4 regions in specific: the extracellular region here, the selectivity filter, the S4, S5 linker, and certain pore lining domains. So this was kind of able to pinpoint certain structural sites that we can start with our actual NMR experiments. So now that we've identified these sites, we can start preparing different protein samples and to do fluorine-19 NMR on. So fluorine-19 NMR isn't exactly very well known, but there's a very good reason why we use it. It's a very ideal NMR probe. It's very, very sensitive. It's almost as sensitive as a proton in terms of NMR. It's very small, so it's structurally non-perturbative. And if you can think about any biological system, you know, there's no fluorine background. So fluorine doesn't exist naturally. So that's one very good reason why we use fluorine-19 NMR. And so there are a couple different methods that we can use to incorporate fluorine into a protein, one of which is covalently. So we basically pick a specific fluorine tag called TET that if we have any sort of cysteine in our protein, the TET will form a disulfide bond to cysteines. And so any cysteine will now be labeled with fluorine. Alternatively, we can use biosynthetic incorporation. So we add a fluorinated amino acid to our growth media so that as we're growing our protein, the bacteria incorporate the fluorinated amino acid. And now in this case, all 25 of our phenylalanines in NaChBac were then labeled with fluorine. So those are two different ways. So using this covalent method uses a technique called site-directed spin labeling. So it's been used extensively, and you can SDSL a variety of amino acids. In our case, we site-directed spin labeled cysteine. So you have to start by identifying all your native cysteine residues, because if you label cysteines, all of the cysteines will be labeled. So you have to create a cis-null mutant, so back mutate any natively occurring cysteines, and then add a novel cysteine to one particular structural site of interest. So in this case, say our natively occurring amino acid was, say, alanine. So we mutate alanine to a cysteine. And then you're going to express and purify your cysteine mutant and label it with your TET, your fluorine-containing tag. You're going to remove any free label via SEC, desalting, and diafiltration. Perform your experiment, but you have to ensure that any free label that wasn't attached to your protein is removed. And then you analyze your data. And maybe it was a great idea, maybe it was a terrible idea, but as I mentioned, these single cysteine constructs, we call them mutants, and in general, mutant is kind of a dirty word in terms of, well, medicine, biology, anything like that. And so when we call them mutants, the reviewers immediately come back and say, whoa, 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 whoa. You're using a mutated protein. How do you know that you didn't perturb the native structure? How do you know that your biophysical properties are the same? But most importantly, how do you know that this functional machine is still actually functional? How do you know you didn't mutate out any sort of functionality? And so to answer that question, because it is always asked, we use 2-electrode voltage clamp electrophysiology. So we take RNA encoding for our mutant, we inject it into an oocyte, and we give it time to express. So the actual cellular machinery of the oocyte will now express our mutant, move it to the cellular surface, and then we can perform these electrophysiology studies where we can actually monitor our protein working. So by altering the voltage for a voltage gating channel, you can then see the current be induced. So, and then to assess whether or not our anesthetics are still inhibiting our protein, we can pass isoflurane or any sort of other drug that we're studying over the oocyte and then hopefully see that our current is indeed inhibited. This sounds great, and it's a very kind of active data collection, but it is not very glamorous. So this is a 9-month pregnant me, 2 days away from being induced, and my boss is hounding me to get this last bit of electrophysiology data we needed to work on this publication. So luckily, I was able to obtain the last bit of data, and then I was able to finally have my kid. So I wasn't going to be able to have my kid without that data, though. You better believe that. So this is kind of the proof of our concept. So we're altering the voltage in this stepwise manner, and we're monitoring the current coming from the actual sodium ions flowing through our protein. So in this case, we start with the wild type NaChBac, no mutants, right? And then we see in black here the channel opening as the voltage is gated. And then in the gray here, we see, in fact, our current is inhibited under the addition of isoflurane. And you can see the 5 mutants that we looked at, indeed, all of them are still activated by our voltage gating. And then, thank goodness, all of them are indeed inhibited by isoflurane. So this is kind of our, okay, we can proceed with our experiment, because if not, there's no sense in using these mutants if they're not functional, because the reviewers are just going to come back and ding us for that big time. So The specific experiment that we used for our NMR study is called saturation transfer difference experiment. And so we incorporate this selectively excitable nucleus, in this case our fluorine, right? And we're going to selectively irradiate that fluorine with our NMR radio frequency. So when we irradiate this fluorine, we give it a certain amount of time to allow that irradiation to transfer to a different fluorine atom. And in this case, isoflurane happens to have fluorines on them. So if indeed isoflurane binds to this specific site, and it binds under certain binding affinities and conditions, the saturation will now transfer from the protein to the ligand, and then you have saturation transfer. So looking at this in a little bit more easily to identify manner. So we have our off resonance spectrum here, and we have a ligand binding to our receptor. And then we have our on resonance where we're saturating the fluorine on our protein. And then if indeed that ligand binds in that location, the saturation will transfer, and then we have a different spectrum in the end. So if we look at our different spectrum and we have no spectrum, that means the ligand is not binding in the site that we incorporated our fluorine atom, okay? So now let's look at this a little. Oh, and I should mention this, sorry. These are the affinity conditions for our ligands. So only in this range of KD will this experiment work. So if something binds too tightly, it won't work. If something binds too loosely, it won't work. But luckily, anesthetics are kind of inherently lower affinity anyways. And so this is indeed met with anesthetic binding. So we started kind of a baseline experiment because we need to prove first and foremost that isoflurane does indeed bind to NaChBac. So we used proton STD NMR. And you can imagine that if protons, there's how many protons in a large membrane protein? Quite a few, right? So if we're saturating all of the protons in a protein and we see saturation transfer occur, you indeed prove that isoflurane binds to the receptor, but because there are so many protons in NaChBac, it's an ambiguous binding location. So we start with this method to say, okay, it makes sense to proceed this because using this experiment, confirmed that isoflurane binding occurs. So now we can look, so this is increasing saturation time. You can see the build-in of the isoflurane peaks. And so that you build your saturation transfer curve from these increasing saturation times. And this is indicative of ligand binding in a receptor using the saturation transfer experiment. So now let's look at our actual fluorine STD. So this is T189, and I picked this site in particular because it's right in the middle of that selectivity filter region, and it's also the narrowest part of the actual pore of NaChBac. So for logistical reasons, it makes sense to start with your longest saturation time, because if saturation transfer is going to occur, it's definitely going to occur at your longest saturation time. So when we perform this experiment and we see this beautiful sort of difference here, so you could see the off resonance spectrum in gray and the on resonance spectrum in black, we sleep very, very soundly at night because this is a very, very good indication that isoflurane indeed binds in this T189 region of NaChBac. So here's the build-in of the isoflurane spectra, and then we have this beautiful saturation transfer curve with about 10% saturation transfer occurring. So we can say pretty conclusively that we know now isoflurane binds at T189. So let's look at all of our NMR data for all 5 mutants. And this is very, very busy. It's way too much to cover in 15 minutes time. And so this, just to make you guys sleep better at night, we did indeed quantify all of this data, so we have errors associated with these binding occurrences. So let's kind of move on, but let's look at this data, all of our NMR data in general, and make some conclusions about where isoflurane is binding and maybe how it's inducing this inhibition of NaChBac. So reminding you of our fluorine saturation transfer, we see definite binding at S129, we see binding at S208, and we see binding at T189, not so much binding at L150 or L179 or in the phenylalanine pore region. So Let's look first at T189 because it's kind of the easiest to explain. So as I mentioned, T189 is at the selectivity filter. So any ion that flows through NaChBac has to pass through this T189 site. And it is also the narrowest part. So you can imagine that if isoflurane binds in this location, how is it going to inhibit ion flow? Well, it's essentially blocked the channel. So now isoflurane's occluding this pore, and now ions can't flow. So that's a really kind of obvious conclusion to come to. For the S129 and the S208 site, it's not quite as easy of an explanation. And so, am I out of time? Oh, I'm sorry. So yeah, binding occurs here, okay? And the inhibition occurs by a restricted pivot motion. So the voltage sensing domain will sense the voltage change, which will induce conformational changes in the pore lining domain of NaChBac. But if isoflurane is stuck in these hinges, now my conformational change can't occur, and I don't get activation of my NaChBac channel. So these are my conclusions. I thought I talked fast enough. I did not. Thank you so much for your attention, and I welcome any questions that you may have. Sorry.
Unfortunately, we don't have time for questions in order to allow the next speaker to start on time, but I'm sure Dr. Kinde will be available for questions during lunch at our poster. Thank you.
I'm sorry. I know you're okay.